Hello, my name is Jeffrey Harvey. I'm a second year here at SF State. I'm majoring in pre-nursing, and the experiment I chose to do was simple diffusion, and I'm going to be using osmosis to demonstrate my understanding of the concept. The reason I'm going to be doing this experiment is because I want potatoes for lunch, and it's very convenient, and I have all the materials I need. In order to do this experiment, you're just going to need a few lab supplies, food scale, 2 bowls, you're going to need 2 tablespoons of salt. I have a measuring cup just for getting 250 milliliters of water into each bowl, and then a potato. What you're going to want to do is just mix your salt water. After that, you're going to want to put your potato slices in. I have 2 slices, both weigh 13 grams each. We're going to let them soak for 20 minutes, and we'll come back and weigh them after. Okay, so now that the 20 minutes is up, we're just going to take the potatoes out, and I'm just going to place them on a plate with a towel just to get all the extra water out. Okay, so while these air dry, I'm just going to talk about what we expect from this experiment. The potato slice that was placed in water should have gained weight because of osmosis, because the salt inside of the potato is already a higher salt concentration than the water itself. And vice versa, we expect the potato placed in salt water to have lost weight because the salt water is now a higher concentration than the salt inside of the potato, which causes the water molecules to float outside of the potato. So slice 1 in water, slice 2 in salt water. Both had an initial weight of 13 grams. Now we're going to weigh the water slice. The water slice now weighs 15 grams. Slice number 2 now weighs 11 grams. What you'll find after doing the experiment is the potato slice that was placed in the hypotonic solution is now more dense. It's also harder to crack open. I know right here. For the one that was placed in the hypertonic solution, super flimsy, easy, crack like that. As we know, hypertonic solutions causes cells to actually shrivel up, and hypotonic solutions causes cells to swell. So a few confounding variables that I found within my experiment relate to the water temperature in the potatoes itself. For the potatoes, each slice seemed to be the exact same size, but the surface area was different by a little bit, and the thickness of each slice was a little off. They both had the same weight of 13 grams, but the surface area and the thickness could have played a huge role. Another thing was the temperature of the water. The water temperature could easily slow down or speed up net diffusion according to Fick's law. Possible sources of error also include the water temperature. As I said before, water temperature plays a huge role in net diffusion. Another thing could be not getting enough salt dissolved into the water. So some other ways that you could build off this experiment or modify it is just using different type of potatoes. You can use sweet potatoes, normal brand russet potatoes like I did. The way you can completely modify it is using gummy bears and having gummy bears in a glass of hypotonic solution and hypertonic, and then measure the weights. Basically, as long as all the concepts are the same and the entire experiment has the same idea, it doesn't really matter what you use as long as your cell has a selective permeable membrane. Thank you guys for watching my week 1 experiment on osmosis. See you guys next time.